Hello biology students, and welcome to video 3 of the evolution unit. In this video, we're going to be looking at the evidence that scientists have for evolution. Okay, so there is quite a bit of evidence out there that supports Darwin's theory of evolution. So we're going to be looking at a couple types today. We're going to be looking at how the fossil record can be used to support evolution, how biogeography can be used to support evolution, how different aspects of anatomy of different organisms and how different organisms develop. And we're also going to be looking at how biological molecules and how they're made up, how that is evidence for evolution. And kind of an important thing to note, like with anything in science, evolutionary theories are best supported when multiple kinds of evidence can be cited. So, the fact that we have so many different types of evidence really suggests that evolution is a strong and sound theory. So we're going to first start with the fossil record. So a fossil is any remnant or impression of an organism that lived in the past. So this could be literally preserved bones or body parts of an organism. It could be a footprint or an impression of an organism in a rock. It could be many different things, and we're going to look at several different examples in a minute. Usually fossils are formed in sedimentary rocks, which form near water, but they can be formed other ways. Sometimes fossils are trapped in amber, or they can be preserved in very, very cold temperatures. There are lots of ways fossils can be formed. So let's look at a few examples. So you can see this fossil is kind of what you think of when you think of fossils. It's literally a preserved skull, so a preserved bone, okay? But fossils can also be different than that. Here I have what's called a cast. So this would be when there's an impression of a rock of something, and then that impression is then filled in by rock or other sediment. So this is not actually the original shell of this organism, but instead an impression of it. Here, this is a impression of dinosaur footprints. So it's a different kind of fossil. This would be fossilized organic matter. So this was the original leaf from however many years ago, but it's been fossilized. So you can see that's different than the other examples. And here I have an insect that's entirely preserved in amber, so the entire body of the insect has been preserved. So all of these things can be used because we can look at the fossil record, which is the sequence in which fossils appear in layers of sedimentary rock, to figure out basically what's the oldest and what's the newest fossils. So the idea is that things deep down have been there for a very long time as sediment piled on top of them. So those are older fossils, where the next layer up would be a little bit younger and a little bit younger and a little bit younger. And then we can use this to kind of sequence how organisms have changed over time. So the fossil record strongly supports the theory of evolution. Basically, the idea is that different layers of rock, which are called rock strata, trap organisms, with the younger strata being on top. And you can kind of look at the historical sequence, so which fossils came first, which fossils came second, and we could use this to kind of track how organisms evolved. So we can use fossils to kind of link early extinct species that we can't study anymore because they're extinct with living species today. So let me give you an example here. So the fossil record often shows transitional species, which is kind of like a species that links an old extinct species with one that exists today. And they often show Darwin's theory of descent with modification. They show how changes are made slowly over time, over many, many generations. So an example is the fact that marine mammals that we have today in the ocean are actually descendants of four-legged land animals. And therefore, if this is true, we should see fossils that have animals with 4 legs, and then animals with slightly shorter legs, and slightly shorter legs, and slightly shorter legs, and each of these in-between fossils would be a transitional species. So let's look at a picture denoting this. Okay, so here is my original organism that is the ancestor of the whale. And you can see, we can look at the fossil record, and we can kind of see, okay, so the organism got a little bigger, and its legs got a little shorter, and then it kind of stayed the same size, but its legs kind of turned into more fin shapes, and then it lost its legs entirely, and then it got much bigger, and then you can see at this point, the whale, which looks very different from the original, but because we have these transitional species in between, we can see how it evolved over time. Okay, so that was the fossil record. So the second type of evidence that we have for evolution is called biogeography. And this is basically the geographic distribution of species. And basically the idea is that depending on where organisms are found in current day, you can kind of figure out who they're most closely related to, who they evolved from, etc. So kind of the quintessential example, we all know about Pangea, how it's, at one point, all the continents were actually one gigantic landmass, and then over time, they separated. Now, because the continents were all one big landmass, if I have a bunch of organisms here, and over time the continents are separating, in theory, the ones on this island shouldn't be that different than the ones over here, because originally we were one species, and they've had, only X amount of time since the islands have separated to evolve separately. So the fact that animals on islands closely resemble species on mainland more than they resemble animals on islands that are very far away makes sense, because the ones that are more closely together most likely broke apart more recently. If they resembled only species that came from similar environments, this would not support the theory of evolution as strongly, because it would not support the idea that things that are geographically near one another evolved from the same common ancestor. So we can also use this same kind of idea to support evolution in different ways. So Australia has been separate from the rest of the continents for a very, very long time. It broke off of Pangea very early. Therefore, it is not surprising that marsupials are only found on Australia, because they have been separate from the rest of the organisms on the planet for a very, very long time. Again, if evolution was not true, marsupials should be found pretty evenly distributed across similar climates. And because they're only found in this one place, and we know that Australia is separated from the rest of Pangea very early on, this supports the theory of evolution. Okay, so that's biogeography. So our third type of evidence type is looking at different anatomical and developmental structures. So the first type of anatomical structure we're going to look at is called comparative anatomy. And this is basically when you take different organisms and compare how their bodies are built, or what their anatomy looks like. And what we're looking for are homologies, or in other words, we're looking for similarities and characteristics in organisms that have a similar common ancestor. So kind of the quintessential example is the fact that limbs in all vertebrates, so all organisms that have spines, look very similar. So let's look at an example of this. So here's the arm of a human, the leg of a cat, the fin of a whale, and the wing of a bat. So these are all vertebrates. And these things are used for very different functions. A human arm is used for grabbing things, picking up things. Cat legs are used for walking. Whale fins are used for swimming, and bat wings are used for flying. And despite the fact that these are all very different functions, they have a very similar bone structure because they all came from a common ancestor. So this fact that these anatomical similarities exist really suggests that they all came from a common ancestor, even though the functions of these limbs are all very different. Okay? So these structures have different functions. So basically, if they have different functions, you would think they would look different. So if they were uniquely engineered to meet their function, they would have different designs, but they don't, which suggests, again, that they all came from a common ancestor and just changed slightly to meet their function. Just like we can compare anatomy, we can also compare embryos of different species. So basically, comparative embryology is where you compare the early stages of development among similar organisms. And again, the idea is because we all came from a common ancestor, we're all going to develop similarly. Okay? Really, this is still an example of a homology. We're just looking at the homology at the embryonic level instead of at the anatomical level. So here's an example. So when humans are developing, the embryo has something called a pharyngeal pouch, which is actually what becomes gills in fish, but in humans it becomes part of the ear and throat. So because both fish and humans have this pharyngeal pouch, it suggests that it's, we're developing from the same way, even though that part of our body becomes different things. So this suggests we have a common ancestor. So I love this picture. So this are the embryos of various organisms at very early stages of development. These are a little bit later, and these are a little bit later. And if you look at the earliest stage, really, you cannot really tell the difference between a chicken, a cow, a fish, a human, a pig, a rabbit, a salamander, or a tortoise. And it's not until later that we really start to differentiate that you can tell them apart. So this kind of shows that we all start developing the same way because we all come from a common ancestor, and over the course of development, we gain more and more differences. So that last one was a cartoon, but here are some actual pictures of embryos. And again, looking at a lemur, looking at a pig, and looking at a human, at the embryonic level, you really can't tell the difference. Again, because we all came from a common ancestor. So the last piece of comparative anatomy and comparative embryology is looking at what we call vestigial structures. And again, we're still looking at homologies. We're still looking at similarities in structures between different organisms. But a vestigial structure is basically a remnant of a structure that used to be important for someone's ancestor, but no longer is important. So it is in the process of disappearing via evolution. So an example, we have an appendix. This is an organ that is slowly disappearing and being selected against by natural selection over time because we don't need it. The tailbone is a vestigial structure that used to be part of a tail that our ancestors have. Whales, even though they swim, they still have the pelvic bones that their ancestors' legs used to be attached to. That's slowly still in the process of disappearing. And flightless birds still have wings or very short wings, which again is also a vestigial structure, something that is slowly disappearing over many generations as it gets selected against. So some examples. So some salamanders slither instead of run, yet they still have back legs they don't use. Whales have hip bones from when their ancestors used to have legs. And boa constrictor snakes also have little tiny hip bones, even though they slither and they don't walk. Okay, I love this picture. So these are little skinks, which are little lizards that kind of slither more like a snake, and they still have little bitty tiny legs that are a vestigial organ from when their ancestor used to walk. So then finally, the last type of evidence that we have to support evolution is the similarity in biological molecules from one organism to the next, okay? So basically, our proteins that do certain jobs in our body look almost identical to proteins that do the same job in other organisms' body. RNA, DNA, proteins, we all use the same monomers. We all use the same building block. So we all use DNA as or most of us use DNA as our genetic material. We have the same genes as bacteria, yeast, fruit flies, plants, which really suggests that we have a common ancestor. And it turns out that you could take your DNA, or you can take the amino acid sequence of some very essential proteins, and you can compare them from organism to organism, and depending on how similar they are, that tells you how closely related you are to that organism. So for example, the human and the rhesus monkey, if you compare a very important protein, that protein is 95% similar between those two species. That means that we evolved separately from one another more recently than humans and mice, who have a bigger difference in that protein sequence. So we share a more common recent ancestor than the humans and the mice do. And you can keep going down the line. The human and the lamprey, we shared a common ancestor very, very, very long time ago because our protein sequence is not very similar, so we've had more time to gain more differences. And what you can do is you can take these differences and you can build kind of an evolutionary tree. So we're talking time. You can see 450 million years ago, there was a mutation that led to two different branches on this family tree. One led to the lamprey, and the other eventually led to the human. Then later on this timeline, I had a mutation that made frogs different, and the other one kind of led to the human. And then even more recently, the chicken diverged, and then the mouse diverged. You can see us and the monkey, we had a very recent common ancestor, but us and the lamprey had a very distant common ancestor. Okay, so basically modern scientists take all of Darwin's theories and all the things he's come up with, and they combined it with what we know about genetics, what we know about DNA synthesis, and then all the different evidence types we have to create the modern theory of evolution. Thanks, everybody, and I'll see you in video 4.